So we were talking about mitosis at the end of the last, meiosis at the end of the last lecture, right? And how it's a special type of cell division that produces egg and sperm. When we talk about meiosis, one of the interesting things that happens in meiosis is that we get genetic recombination happening almost immediately, right? So remember that meiosis is two cell divisions. So we have meiosis 1, which would have prophase 1, metaphase 1, anaphase 1, and telophase 1. And then immediately those cells undergo another second cell division. So in prophase 1 of meiosis 1, we get the genetic exchange between replicated homologous chromosomes. And that is shown here, okay? So what is that called when we exchange genetic materials? You have that in your notes. What is that specifically referred to as? It is a form of genetic recombination, but when they exchange, it's called crossing over, right? So crossing over is the exchange of genetic material between replicated chromosomes. Do you have that in your notes?
We have it as prophase 1.
Prophase 1 is crossing over, right? So that's just crossing over. Okay. So that is a form of genetic recombination. Now, what is the cost to this genetic recombination? Mutations. What is the benefit? Variation, right? So variation in the offspring, and that variation might be really important in terms of like allowing us to keep up with all the pathogens and the microbes that are co-evolving with us, right? So we need to be able to evolve resistance to particular diseases or illnesses that might be in our environment, okay? Okay, so when we look at the different stages of meiosis, one of the things that differs between mitosis and meiosis is what is referred to as metaphase 1. So we looked at prophase 1. Metaphase 1 is also very different. So whenever you see a 1 behind something, then you know that it's referring to meiosis, not mitosis, because there's only one cell division in mitosis. So metaphase 1, you'll notice here that we have replicated pairs of chromosomes lining up in the middle of the cell. So I'll put they line up in the middle of the cell. Okay. So notice how when they are pulled apart, the sister chromatids are not pulled apart. So the chromosomes after meiosis 1 are still replicated, right? Because they have not been pulled apart. Also notice that we could have them arranged in different ways. So here, the blue and the reds are together, and they separate together, right? During meiosis 1. Here, they're separate. They're actually put together in a different arrangement. And so we can also have genetic recombination because the way that they are arranged is random, right? So the arrangement is random. So this also increases the genetic variation. Now, remember that when we're looking at the way that chromosomes behave in these cells, you have to remember that we have actually 23 pairs of chromosomes. This is just showing 2 pairs of chromosomes, right? So if you can imagine, you know, all the different arrangements that they're lining up, if there's 23 sets of them, the way that they line up could be very, very disordered, right, from one another, right? So there's a huge amount of variation that can be produced because of that. So notice how in this instance, like this egg or sperm, this gamete, only got blue chromosomes. So like that would be all the father, right? So this one only got father chromosomes. This one only got the chromosomes that were from the mother, right? And so from that person's mother, right? So, but then notice in this particular arrangement, right, there is more variation, okay? So that the way they line up is important in the way that they separate, okay? So that is metaphase 1, and then at the end of meiosis 1, the chromosomes are still replicated. So the whole point of meiosis 2 is to separate them. In meiosis 2, the chromosomes are separated. And then at the end, so that at the end, the chromosomes are unreplicated. Okay. So you notice that in metaphase 2, instead of them lining up, there's no longer pairs of replicated chromosomes. So they line up like this, right? And then they are pulled apart. The sister chromatids are pulled apart. And so you'll notice that when we look at the gametes that are produced, there's one big chromosome and one little chromosome. And what that means is that there are no pairs. So notice how there are no chromosomes that are of the same size and the same shape. There are no pairs. And the whole point of this process of meiosis is to have the genetic material so that when the egg and the sperm come back together, the genetic material will be a full complement and will not like be doubled. Like if you just had a two diploid cells come together, then it would produce a cell that had 4 times the genetic information, right? So we need to have the genetic information, and we also need to produce variable gametes. Okay, so that's the significance of meiosis. And this is, we're going to talk about this because we are going to be moving into Mendelian genetics. And when Mendel was doing his work on peas in the 1800s, he had no idea what even a chromosome was. But now we can make sense of his experiments because of the way that the chromosomes behave during the production of egg and sperm. Okay. So in your book, they show you the difference between mitosis, which is just one cell division, right? And then we get the difference between meiosis, which is meiosis 1, and meiosis 2, and then it produces cells that have half the genetic information. Okay, so you're still a little bit fuzzy on this. You might want to just take a look at this diagram. Okay. Okay, so there is a difference between the egg, the production of the egg, and the production of the sperm. And the whole difference, they believe, is based upon the fact that the egg needs to have all of the nutrients provided to it that the developing embryo is going to need early on before it gets nutrients in another source, okay? So oogenesis is egg production. And something really interesting happens during egg production. So we have a diploid cell that is going to undergo meiosis. So this is a diploid cell. And so it is going to divide. Now, something happens where one of the nuclei that are produced gets just a tiny bit of cytoplasm, and the other cell gets almost all the cytoplasm, okay? So notice that we now have what is called unequal cytokinesis. So this is like in meiosis 1. This is called unequal cytokinesis. Okay, so unequal divvying up of the cytoplasm. Now this nuclei has genetic material, it's perfectly fine, it just doesn't have any nutrients in it, right? So this is just discarded. And does anybody know what that is called? It has a name. Anybody know what it's called? It's called a polar body. Polar, like polar bear, body. Okay, and it's usually called the polar body because it actually kind of stays adhered to this little cell, and we're going to watch a video of that. So you'll see one big egg, right, and then you'll see just a little nuclei off to the side, and that is the polar body. Okay, so you think, oh, that's pretty weird, and that's kind of wasteful, right, to get rid of half of your genetic material. And then another polar body can be produced here, right? So we have the nuclei, and then we have the cytoplasm, and then we produce a large egg, okay? So this would be in meiosis 2. So we actually produce 2 polar bodies. Sometimes actually produce 3, but generally it's just 2, okay? So this egg has all the mitochondria. Remember we talked about mitochondria being inherited from your mother, right? And it has all the nutrients. And so I've mentioned before that that's how we define, if you were to go to another planet, like for example, and they had sexual reproduction, that's how you would define males and females, right? The females produce the large gametes, and the males produce the small gametes, okay? So in spermatogenesis, this takes place in the testes, right? And spermatogenesis is the production of sperm, and this is equal cytokinesis. Right? Equal cytokinesis. So what that means is that we get from one diploid cell, we get 4 haploid cells. Notice up here, we started out with 1 diploid cell, right? And we only got really 1 haploid cell. So in oogenesis, we start out with, so right up here in oogenesis, you start out with 1 diploid cell, right? And you get 1 haploid cell. And that is the egg. Okay. For diploid cells, these are the sperm. Now, in order for the sperm to be competitive, they have to be fast, right? To get to the egg, the first one, it has to be pretty strong to survive all the hostile environments. And so they actually discard most of their cytoplasm so that they can be faster and lighter, right? And so that's the sperm. Okay, so we're going to watch a short little video that specifically talks and shows images of meiosis occurring in the ovary and the testes, and then it talks about fertilization in humans and how that is accomplished. Oops, where's the volume? It's there. Let me just check the volume on the computer. There we go.
Okay. A woman is born with all the egg cells that she will ever have. There are about 1 million of these primitive cells called oocytes. By puberty, this amount is reduced to 400,000. Over the course of the fertile years, only 400 eggs will grow to maturity. 400 chances to make a baby. Here, with the addition of a ring of supportive nurse cells, an oocyte has developed into a follicle. Several follicles are created each month, but only one continues to develop into a mature egg. Every oocyte and every sperm carries the unique combination of genes that make up our inherited characteristics. Here, the egg is collecting all of its genes in a little sac. The sac is then broken down and dissolved. The gene pool divides in half, and one part is pushed out of the egg.
That's
Out of
the
46
polar body
chromosomes,
right there.
23 are left, the mother's share of a child's biological inheritance. The eggs are held in the woman's ovaries, the almond-shaped organs connected to the uterus by the fallopian tubes or oviducts. The man's reproductive cells are found inside the 300 feet of coiled passageways within the testicles. At birth, they are immature, lacking tails. Beginning at puberty, the sperm mature at a rate as high as 1,000 a second. The dark blue rods are the 46 chromosomes, the man's genetic identity. But as this primitive sperm divides, 2 cells are created, each carrying just 23 chromosomes. The sperm now grow tails to help navigate the long trip to the egg. When the 23 chromosomes contained in any one of these sperm combine with the 23 chromosomes of a mature egg, the result will be a new combination of 46, the blueprint for a completely new human being. In the ovary, the monthly process of ovulation is about to begin. First, a single follicle expands like a bubble on the surface of the ovary. The bubble ruptures, and the egg's nurse cells are propelled out, seeming to lay out a protective carpet for the egg. These are the first pictures of ovulation ever photographed. The egg is gently caught by the frilled edges of the fallopian tube and led into the narrow channel leading to the uterus. At first, tiny muscle contractions in the fallopian tube push the egg along its way. But then the cilia take over, millions of microscopically fine hairs that slowly sweep the egg along. So slowly, in fact, that it takes 4 days for the egg to reach the uterus, longer than it can survive on its own. If a baby is to be made, the sperm will have to join the egg here in the fallopian tube during the brief period when fertilization is possible. The sperm arrive on the scene during sexual intercourse at the moment of ejaculation. At male orgasm, typically several hundred million sperm are ejaculated into the woman's vagina. They have a common goal: get to the egg and fertilize it. Only one, if any, will succeed. Millions of sperm die at the first pass, destroyed by the vagina's protective acidic environment. The survivors continue up to the cervix, the entry gate to the uterus and egg beyond. If a woman is not ovulating, the secretion barrier of the cervix spells doom for the sperm. There is zero chance of getting past it. Folds in the mucous membrane trap the sperm, and they struggle to escape for up to 3 days before they die. During a few days of ovulation each month, with the mature egg waiting, the impenetrable gate of the cervix opens ever so slightly. Sperm are still trapped by the thousands, but a few find the microscopic channels created in the secretion barrier by the release of estrogen. The surviving sperm are halfway home. They push on through the uterus up to the entrance to the fallopian tube, Of the 300 million that began, perhaps a few thousand remain. Now, within striking distance of their target, they prepare to do what they've come for: fertilize the egg. Just past the fallopian tube entrance, the surviving sperms suddenly stop and rub themselves against the cilia. It is possible that this is the moment when a covering of protein molecules on the sperm surface is stripped off. With the covering removed, the sperm, for the first time, are ready to deliver their chromosomes, fertilizing the egg. So close and yet so far. Of the thousands that make it as far as the fallopian tube, only a few hundred reach the egg. When they get there, they encounter the final and most imposing barrier of all, the egg's protective shell. On the cellular level, this dense cement-like ring is the equivalent of a castle wall 30 feet thick. Special measures are called for. Arriving at the egg, enzymes carried in the sperm's body rush to the surface and form a chemical battering ram. The chromosomes remain in the sperm head, but the tail, powered by energy capsules called mitochondria, changes its motion from regular waves of movement to quick, strong, whip-like lurches. An amazing effort, but without the cooperation of the egg, it probably wouldn't be enough. The support cells that now make up the wall of the egg are changing shape, weakening the wall just enough to draw the sperm in. The sperm keep trying, the explosive energy of their tails driving them along. Most of the remaining sperm die in this last attempt to penetrate the egg. Finally, success. A single sperm has broken through. Instantly, a burst of electrical activity changes the surface of the egg, making it totally impenetrable. 300 million sperm set out. One made it to the finish line. The sperm continues to move for a short time while inside the egg, and then its tail drops off. The sperm's head slowly starts to swell until it is 200 times its original size, preparing to combine its genes with those of the egg. The two large round structures hold the ingredients for a new human being. Half the genes from the woman, half from the man. Now, the moment of fertilization. The two pools of genes appear here as the two interlocking circles. They fuse with immense power. United, they dissolve into the egg, and the blueprint for a new individual is set. A combination of inherited characteristics unique in all the world has been created. Almost immediately, the egg divides itself into two identical cells. These daughter cells are the first of the billions to come that will culminate in the birth of a single new human being, or maybe more.
Okay. So what they were referring to there as, or maybe more, would have been identical twins, which we'll talk about in a minute. Okay, are there any questions about that video? So you did see the polar bodies, even the polar bodies were even still present on the egg when fertilization took place. So let's see if I can rewind that a little bit. Okay, so this is why they're called polar bodies. This is that they're kind of still stuck, right? And these are, there's cells around the egg that were what are referred to as the nerve cells, that when the egg was ovulated, they kind of just stay and they form what they call the corona radiata, which is a crown of cells that help in the support of the egg. The egg will eventually break free from that as it develops into an embryo. Okay, so let's talk about sex determination. So in mammals, we have a specific type of sex determination that is due to the presence of specific chromosomes. And so if we look at females, what combination of sex chromosomes do females have? XY, YY, XX, what combination? XX, right? So this is a female sign. So the female has 2 copies of the X chromosome. So they are said to be homogametic. And what that means is that they can only produce eggs with the X chromosome, right? So the males in mammals are XY, and they are heterogametic. Meaning that some of the sperm have the X chromosome and some of the sperm have the Y chromosome. Okay. So if we look at the ratio of predicted egg and sperm, if you think about a female, she has 2 copies of the X chromosome, and by chance, we would predict that half of the eggs would have 1 copy and half of the eggs would have the other copy, right? In males, so I'm going to draw this down here, the males, they are XY. So you would predict that half of the sperm would have the X chromosome, there's an X right there, and half of the sperm would have the Y chromosome, right? And so this is what they mean when they say that the males are what determine the sex, because it's the chromosome in the sperm, the sex chromosome in the sperm, that determines whether the baby is going to be male or female. So on average, if we did like a little combination here, which we'll talk about this is called a Punnett Square, okay? We can combine the egg and the sperm here. That would be a female offspring. This would be a female offspring. This would be one X and one Y. That would be a male offspring. And this would also be a male offspring. Okay. So on average, we predict that half of the babies would be male and half of the babies would be female. One of the really interesting things that we do find is that more baby boys are conceived. So it is thought that this Y, the sperm with the Y chromosome, actually might be faster. So because it has less genetic material, the X chromosome is much larger. So if we looked at an example, this is the X chromosome, and then this would be the Y chromosome. So this is an X and a Y. And so the Y chromosome is much smaller. And so perhaps that would make this sperm a little bit more faster, so that we might have more boys conceived. However, we'll talk about this, but there can be a higher, or more of a problem genetically, because they have only 1 copy of all the genes on the X and only 1 copy of all the genes on the Y. So if there's a bad gene on the X, they don't have an extra copy. And so actually baby boys also tend to be more prone to spontaneous abortions. So miscarriages are going to happen more frequently in male offspring than female offspring. Okay. So I want to show you a little video that talks about some other kind of interesting things about males and females, and it appears that we can adjust the sex ratio of our offspring in response to what is going on in the environment. So let me just video up. Okay, so this video is quick, so you kind of have to pay attention. It's not a slow video. Oops. It's 2.5 minutes, but it explains a lot. Okay, let me turn this slide off.
Assume that the sex of their child comes down to a flip of the chromosomal coin, with an equal chance of having a boy or a girl. But in reality, the odds aren't even. For every 100 girls born, the world gains about 106 baby boys. Currently, that skewed sex ratio comes out to roughly 10 million more baby boys than girls born worldwide each year. There are some countries where human meddling stacks the odds even higher in favor of boys, but that doesn't explain the fixed odds everywhere else. The intrinsic boy-girl ratio is rigged by Mother Nature. In fact, it's even more rigged than birth rates show. Human conception results in about 150 male zygotes for every 100 females. But there's a rather tragic reason for this big biological boy bias early on. Male fetuses are much more likely to be miscarried or stillborn than female fetuses. And boys that do make it out of the womb suffer more fatal diseases, take more mortal risks, and fall prey to more violence than girls. So by the time kids grow up and reach baby-making age, the ratio of males to females is just about 1 to 1. But the likelihood of a boy even making it to birth is also influenced by his mom's living conditions during pregnancy. For example, when a massive famine struck China in the 1960s, the relative likelihood of having a son suddenly dropped until the famine ended. And male Americans born to billionaires seem to have higher than average odds of fathering sons. Somehow, female biology suppresses boy survival in the womb during tough times and boosts it when times are good. We've seen the same pattern in other mammals too. When resources are scarce, mothers give birth to fewer males than normal. When resources are plentiful, they bear more. The best explanation we have for this has to do with sex, the other kind. In biological terms, the whole goal of copulation is to reproduce, to pass down your genes to someone who will someday pass them on again. Female offspring are almost guaranteed to reproduce, famine or no famine, because male mammals are pretty much always willing to mate. Males, on the other hand, have to compete for mating privileges. A well-nurtured hunk has a good chance of mating with lots of females, while a male weakened by famine might not score at all. So male offspring are a bigger risk in general. At all stages, they're more likely to die. And even if they live, they might not reproduce. But when times are good, boys' potential to father lots and lots of babies make them a biological risk worth taking. Thanks to everyone who supports us on Subable. Without you, we literally couldn't continue to make these videos.
Okay. So if times are good, are boys more likely to be born or are girls? Boys, okay? And that's because the problem of having to compete for access to mates, and there's also the problem that they are more likely to die, right? So if the mother, if her physiology senses that the environment is good, then she's going to be more likely to produce boys. If her physiology senses the environment is bad, she's going to be more likely to produce girls. Okay? So there is a bias in sex ratios. Any questions about that idea? Okay, so I put mammals up here because sex determination in other organisms is different. So for example, birds are exactly the opposite of this. They actually have a chromosome called the Z and the chromosome called the W, and it's the female that is ZW and the male is ZZ, right? In reptiles, oftentimes it is environmentally induced. So if the eggs are hot, then they tend to produce males. If the eggs are incubated in a cooler temperature, they tend to produce females. So in some species, it's not determined by a genetic difference at all. It's only determined by the environment and how genes are turned on and turned off that determine the difference between males and females, okay? And then maybe we'll talk about bees, honeybees, which are really interesting, okay, examples. Okay, so one problem that we have is that we can get errors in the way the chromosomes separate during meiosis. And so one way that we can get an error is through what is called non-disjunction. So this means that the chromosomes do not separate properly in meiosis or during meiosis. And so this can lead to a problem which is called aneuploidy. Okay, so what this term means, if you break it down, A means without, and they put an N in there because A and E would be hard to pronounce. So this is without. U means good, a good. Ploidy means number, like how many copies. So this is without a good number of copies. Okay. So when we look at our genetic material, we can say that 2n is normally equals 46, right? So if we have 2 copies of our chromosomes, this is said to be diploid, right? And so 2 times 23 equals 46. So n is the number of chromosomes, right? So we can have problems where we have like 2n plus 1. So we could have 47 chromosomes, for example. We could also have 2n minus 1, so that we would have 46 chromosomes. Humans and mammals in general cannot withstand more than 1 extra copy of a chromosome, or if there's just 1 or 2 examples of the 2n minus 1. Right? Actually, there's only 1 example that I know of. Or, oops, that should be 45. Where they have 45 copies, right, rather than 46. Okay, so this is the good copy. So I'd say that this is euploidy, right? So these would be aneuploids. So sometimes we can detect whether or not there are an incorrect number of chromosomes by looking at a karyotype. So a karyotype is a picture of all the chromosomes. And so if you suspect maybe a child might have a particular illness or disease, you could do a karyotype on them. The karyotypes are probably more commonly done on developing fetuses to determine if there is a genetic problem that they can visualize looking at the chromosomes. So when we look at an image of a karyotype, okay, here we can see that if we looked at all of the chromosomes, we could pair them up based upon their size and their shape and their staining pattern. And then this individual would have 3 copies of 21, chromosome number 21. So what is that referred to as? We have an extra copy of chromosome number 21. It's a trisomy of 21, but that's commonly called Down syndrome. No, is it Down syndrome? Down syndrome. Is it with a Downs? I can't even remember. It is with a apostrophe S? Yeah. Thank you. Okay. So that's trisomy 21, Down syndrome. Is this individual a male or female? You can tell that from the karyotype too. Female, right? So they have 2 copies of the X chromosome. If they had a Y chromosome, they wouldn't have this, right? And then they would just have a little Y chromosome like right there. And so then that would be a boy. But this is definitely a girl because there are 2 large sex chromosomes. So when we look at how this could possibly occur, how could we get an extra 21st chromosome? We could go back to that idea of the chromosomes not separating properly. So this would be meiosis, and this just shows non-disjunction. And so it could occur in meiosis 1. So notice that these chromosomes did not separate properly. They all went to one of the eggs, right? And then what happens is that you get an extra chromosome here in those eggs, but these ones are missing a chromosome, right? So if a female produces an egg with an extra 21st chromosome, she could also be producing an egg that's missing a 21st chromosome, right? But if you have one 21st chromosome, you cannot survive. And so you would be spontaneously aborted, right? And so this can also occur in meiosis too. So probably the reason why, when we look at this, why we see babies being born with an extra 21st chromosome, oops, I went the wrong way, an extra 21st chromosome, rather than saying an extra first or an extra 10th, right? Sometimes they can be born with an extra 13th and an extra 18th. Those are survivable. Is that this chromosome is really small, right? So it's only an extra copy of a relative small number of genes, right? But non-disjunction can also occur where you would have an extra copy of a first chromosome. But that would be such a big, mistake that you'd have an extra copy of all of those genes that you would die, right? So you would actually never see a baby being born with a copy of an extra first chromosome, but it could happen just as much as it can with the 21st, right? And you would never see them with just one copy of the 21st chromosome, right? Because that is also lethal. So when we look at embryos, they estimate that 33% of all embryos produced are spontaneously aborted. And most of these are before the female even knows she is pregnant. So it could be that she's a week late on her period, or maybe 2 weeks late, right? Or it could be that she tests positive and then she has a miscarriage, right? So what this means is that there's lots of mistakes that occur in meiosis that could lead to a lethal error that could mean that the embryos are not able to survive, okay? So we have lots of mistakes during, lots. I'll put many mistakes occur, but these do not survive. Okay. So the embryo, interestingly enough, has to prove itself healthy enough to maintain the lining of the uterus. And it does that by producing a hormone called HCG. And so if the embryo is not able to produce like huge amounts of HCG, which is the chemical that they use to detect pregnancy and over-the-counter pregnancy tests, then the embryo is aborted. So it's screened out. So if the embryo is not healthy enough, it will be screened out. Okay. Some embryos can't even implant, right? They have to actually produce enzymes to implant in the lining of the uterus. Okay, so another thing that we could have is that we could have a defect where we could have a breakage in a chromosome. So another chromosome abnormalities can be detected. So we can detect changes in chromosome number and aneuploids, and then we can also detect changes in chromosomal abnormalities. So chromosomes can break. And you can get deletions, right? So you can get a chromosome being broken, and you can get it deleted. And so a person looking at this, you're like, I don't even see the difference between those chromosomes. But the darkly staining region at the top is missing. So it actually has a deletion. And this is chromosome number 5. So a deletion on chromosome number 5 is one of those genetic mistakes that actually survives, so that we actually see it in being born, a baby being born with this. And this is a disease that is called Karyotype. which means in French, cry of the cat, because of the way that they sound when they're babies and they cry. They sound like a kitten crying, right? And so you can see that this is actually one of the most common syndromes caused by a deletion, right? And so it affects between 1 in 20,000 and 1 in 50,000 babies. And unfortunately, it's a really bad genetic error to have. And so it causes all kinds of problems in people that have it. And so they have usually small at birth, they have respiratory problems, their larynx doesn't develop correctly, so that's where their voice box is, and so that's why they have that signature cry, right? And then they have microcephaly, which you hear a lot about in the news because the Zika virus also causes microcephaly. And so they generally don't tend to live a normal life expectancy. So they generally tend to die of that particular syndrome, okay? Okay, so I'll stop there for today, and we're gonna talk about Mendelian genetics. Don't forget to complete the quizzes. Is there 2 quizzes this week? Yeah. 2 quizzes this week on the online homework. Oh, there's a box over there too, that you can use this one. So over there on the table, if you want to drop off your clickers over there, or if you come in this direction.
Welcome back to high school, folks. Yes, hated that time of my life.
You hated genetics?
I hated sex ed.
Oh.
Genetics, actually.
I can stand genetics. I'm a rancher. I need to understand genetics. Genetics was fun. Sex ed, not so much.